All right, we are here. It's 1 o'clock. Time for Bio 1. All right. Ooh. Okay, so obviously a lot of changes have happened with the school, so hopefully we're all notified that the district has proclaimed that we're going to be doing remote instruction for the rest of the term. All right, so this is definitely our new normal. As in completely closed, not entirely. There will still be some basic campus services that will remain available, like access to computer labs, library, and certain basic campus operations are still happening. But for the most part, everything else is being done by remote. So that's kind of where we are. Uh, I've got, again, kind of a detailed plan that we're going to be operating in terms of be looking for some more laboratory resources and things to get through the remaining labs. The group research project on the yeast, I'm just going to have to cancel all of that since there's no really an opportunity for you to work together under all the different restrictions and things like that, plus just good old common sense is that we shouldn't be meeting with too many other people more than absolutely necessary. So that's kind of where we are. Right. Exams are just going to continue on kind of the way we did exam 2. We'll just make 2 and 3 also take-home type exams. And again, I hope to sit down and really start working and grading those and getting those back to you definitely by sometime next week. So with spring break, I'll have a little more time to work on that kind of stuff. We still are going to adhere to the spring break schedule, so there will be no classes next week and there will be no assignments due during the next week. So hopefully that covers all of the basics. Question is, do the exam questions still come from the study guide? Yes, they do. So it definitely behooves you to really take the time and as we go through the lectures, mark up your notes as to what seems to apply to each study guide element, and then start working on crafting responses to those ahead of time. Ultimately, the real learning happens when you guys are engaging the material and you're trying to process all this information into your own words and your own explanations. So if you're doing that on your own ahead of time, and then I load up an exam and you've already got a bunch of stuff already typed out and ready to copy and paste, I'm really okay with that. The important thing is that you're trying to understand the material, and the time frame on it isn't necessarily as important. Certainly, the practical reality, kind of like we joked a little bit about, learning the Krebs cycle, right? And, it's the cycle is learn the Krebs cycle, forget the Krebs cycle, and then learn the Krebs cycle again and forget the Krebs cycle. A lot of your undergraduate education kind of goes through that mode where you learn something, but then you forget it until it's time to learn it again. And hopefully the second time you learn it is easier and you keep going back to it. And you can get to the point where it's really, really ingrained in your memory and you really understand what's going on. So a lot of what we do here is really just kind of the first steps of your learning process. And hopefully you take what you have from here and you build on it into the future. So, yeah. Okay. Oh, new question. Do we have to turn in the drawings for your mitosis, meiosis stages with the lab questions? Yeah. I think that's fair and easy to do. It's the easiest thing for me to kind of assess that you are kind of visually understanding these processes. And again, hopefully by the time we get done with today's lecture, you will be able to complete the last set of illustrations for meiosis. We have some really good models for you to work yourself after. So yeah, I think you guys can go ahead and scan in those pages. All right. And then is another question. Is it acceptable to use Photoshop-like programs if we've completed those drawings? Let me think about that. But again, it seems reasonable if you're able to go with something that is directly electronic rather than doing it on paper and then trying to scan it in with some kind of a scanning app or program or something. Sure. I think that would be a fair equivalent. Oops. Hanging things around here. All right. Okay, anybody got any other questions out there before we kind of get into and get the lecture going here? All right. Okay, let me just get this. All right, and start our closed captioning while we're going along. All right. So if nobody has any additional questions out there, all right, then let us start off with this topic here. All right, so chapter 13 is a topic of meiosis and relating this to the sexual life cycle, the way that most eukaryotic organisms perpetuate themselves. So let's kind of start with kind of really the background problem that this topic is really going to help us to understand. And so the problem is kind of questions of there's a certain amount of sameness among organisms when they reproduce, and yet there's also variation when they reproduce. And it kind of starts out like this. Okay, so as we talked about in mitosis, when a single-celled organism reproduces, it reproduces using a mitosis or mitosis-like process, and as a result of it, the offspring are completely identical to the parents, especially kind of on that genetic level. All of the information that were in the parent cell have been faithfully copied, and then the copies were split and separate from one another, and then the cell itself divides. And so as a result, all of the offspring are completely identical to the parents. And that's really what mitosis does. Mitosis maintains that genetic continuity. So that's really, really good. And for a lot of organisms, that works really well, because often when single-celled organisms are surviving in their particular environment, they're probably very well adapted to their current circumstances. And so making more offspring cells that inherit those same adaptations makes sense. You know, if it ain't broke, don't fix it. All right, so that's really, really good, and it helps organisms to produce lots of offspring in a relatively short amount of time. But if they're all genetically the same, and you have a large population of organisms that are all kind of genetically clones to one another, what happens if, when inevitably that environment starts to change and the adaptations that the population used to have, or that they currently have, are no longer adequate to new environments? And so this is an okay strategy when you have a very stable kind of environment, but if the environment is not stable, there are going to be issues. Now let's also contrast this with the approach of other organisms. When organisms use sexual reproduction, they make offspring that are not identical to either one of the parents. I mean, think about that. For those of you who have siblings, right, you have biological brothers and sisters. Do you look identical to your brothers and sisters? Probably not, right? Unless you happen to be an identical twin, and there's reasons for identical twins to have such a high degree of similarity in their appearance and traits and features. But otherwise, we're all very different, you know. And also, none of us is an exact duplicate of either one of our parents, are we? You know, you look carefully, you will see traits of both parents in the offspring. One of the things I used to do, it's gotten a little dated in time, but it's still kind of fun to look at, would be looking at a family portrait of mine with my wife and I and our first child, my son Stephen, and when he was only a few months old. And yet, even at that early age, you could see that there are definitely some traits that he has that are mine, and he has other traits that are more from his mother. And so we're kind of a mix of characteristics between the two parents. So, but that makes him unique and different. Right? So the big question is why? Okay, why are offspring different in appearance than their parents? Right? To answer that question, we need to take a little bit of a closer look, though, at the idea of chromosomes. All right. So we'd already talked about chromosomes in the context that these are discrete pieces of DNA organized by proteins, and that during interphase, all the chromosomes are in a very loose, spread out kind of a state that we called chromatin. All right. So, and then mitosis was all talking about how in order to separate the copies of the chromosomes from one another, they have to coil and tighten up and to become these visible thick rods that are called chromatids. And again, we use the terms monads for the individual chromosomes, and when they're joined together, we call them a dyad. All right, so that's a quick little review there. All right, but we need to take a little bit more of an understanding of chromosomes in eukaryotic organisms. All right, so first off, all eukaryotic organisms, they differ in the number of total chromosomes that they have. So, for example, humans, as you probably know or have heard, have 46 total chromosomes. Other species have different numbers, so like fruit flies, what we're going to learn more about in another couple of weeks, they only have 8 chromosomes. Okay, and so that's sort of one of the differences from one species to another, although it's really not the most important difference. The most important difference are the actual genes that are on the chromosomes, and the kinds of genes and how they're activated is really what makes one species different from another. But certainly this is a facet of difference among species, is just having different numbers of chromosomes. But what's interesting about our chromosomes in sexually reproducing organisms is that every chromosome actually has a partner, all right, or sort of a twin, okay, or maybe at least a near identical twin or a sibling. And the reason why they have these twins is that, or what makes them twins, is they have the same number of chromosomes. I'm sorry, I'm getting ahead of my thought process there. They have the same genes on them. So, an example, you might have a chromosome, and on that chromosome is a gene that helps to determine your eye color. And then you can have a separate gene that determines how tall you're going to grow. And you have another gene that determines what types of proteins you're going to use to construct cells. All right. So you've got all these different genes. And then you have another chromosome that has the same genes on them. All right. So these paired up chromosomes are referred to as homologous. Okay? So again, the prefix on that, homologous means, homo means to be the same, all right? So identical or nearly the same, okay? Logos is like from the Greek word logos, meaning word, kind of describing information. And so these are chromosomes that contain the same information. So we call them homologous pairs. So ways you can recognize homologous chromosomes, well, you can't really see them except when the cell has condensed all the chromosomes during mitotic cell division, and during metaphase in particular is when they all can kind of become visible within the cell. Again, if you remember studying the pictures in the slideshows during for the lab video, you were able to visibly see chromosomes inside of cells. All right. There's even techniques where you can take isolated cells that have been allowed to progress into metaphase, and then you can lyse or break open those cells to release the chromosomes from all of the membranes, and then you can spread them out onto a microscope slide. You can actually then look at these individual chromosomes under a microscope. And if you do that, then you begin to see that the chromosomes really do come in pairs, because you'll see pairs of chromosomes that are the same size. And since they were in metaphase, the monads are connected to each other by a centromere. The placement of the centromere along the length of the monads, or basically its position in the whole chromosome, they're in the same locations, and different chromosomes will put that centromere in slightly different spots. Some will have it perfectly centered among the chromatids. Others will put it a little off-center, and so one side will look longer than the other. So here's kind of an idea. Here's kind of an over-representation of what they might look like. The colors are completely artificial. That's just for decorative purposes here. But when you stain chromosomes during metaphase, there are certain stains that will bind to some parts of the chromosomes and not other parts, and you kind of get these alternating light and dark patterns. And so that's where these colors are kind of represent, or the different bands that either be lighter colored or darker colored. So a homologous pair then would be the same size in the pair, position of the centromere is the same in the both, and the overall banding pattern is completely the same. But what's really happening on a genetic level is that the location for a particular gene is in the same place on the chromosome for each one's in the homologous pairs. So again, these blue bands down here might be the location of a gene that helps determine height. And then up here, these green bands on the top, these might be the location of a gene for a person's hair color. Right. Now, again, the other thing that you'll notice here is that the chromosomes are these dyad type chromosomes because you've got two monads joined together. And so this becomes an aspect of understanding how what we're going to do with these chromosomes when we get into the topic of meiosis. All right. So what does this have to do with sexual reproduction? Well, in order for organisms to reproduce sexually, okay, we have to deal with the fact that the normal cells of the organism all have these homologous pairs, and therefore you have two sets of chromosomes. And there's a term for that. We call this being diploid. Okay? So diploid just means that the cells of the organism all occur in homologous pairs. So you got two of everything. All right? So this is actually kind of a good thing for life, because later on in the term we're going to talk about the subject of mutations and what happens when genetic information gets corrupted or altered. All right? The nice thing about being diploid is that in the event that one copy of a gene becomes mutated or corrupted, cells still have an alternate copy that still provides the functionality of the gene. So in other words, it's kind of like having backups. It's like what we try to do with all of our computer files and information. If we have really important information, we don't just store the file on our hard drive, on our desktop, or on our laptop computer, because what happens if the hard drive crashes, or what happens if the file gets corrupted? But if you have a backup, so if you store all of your critical files out on a cloud or on a separate thumb drive that you keep in a safe location, then you can still have functionality. You can still get to the program, or you still get to the file, even though one copy of it got damaged. So kind of biologically, that's the benefit of being diploid. It provides backup copies to everything. All right. Here's the problem, though. If we used ordinary cells to then combine with the cells of another individual, and that's really what sexual reproduction is all about, is using the genetic information from two individuals to create an offspring that is kind of unique. If individual A used a diploid cell and individual B used a diploid cell, all right, so you got two copies of every chromosome from individual A, two copies of every chromosome from individual B. Now you put those together. Your offspring now has 4 copies. And it turns out that living things generally don't do well with lots of extra chromosomes. Too much genetic information turns out to be a bad thing. It gets too difficult to regulate all of those genes at the same time and keep a balanced functioning to the cell. Now, it does occasionally happen, and we'll talk a little bit more later in another chapter about instances where extra chromosomes get donated due to mistakes or errors, particularly in the cell division processes leading up to sexual reproduction. So, I mean, they can happen, and life can sometimes overcome those barriers, but very frequently, that's bad. So, in order to reproduce sexually, reproductive cells, the egg and sperm, only have one set of chromosomes in them. And that condition is referred to as being haploid. Kind of like having half the number of chromosomes that the organism usually has in its cells. But egg and sperm cells, we're going to cut that number of chromosomes in half and try to make it so that there is only one copy of every gene in that reproductive cell, so that you have one copy of genes from individual A, one copy from individual B, bring them together, now you get two copies. All right. So these specialized cells, if we're not worried about which parent they came from, we call them gametes. So gametes is kind of a gender neutral term for a reproductive cell if we don't need to specifically call it an egg or a sperm cell. Obviously, sperm is the term we use as the cells contributed by males, and eggs are the cells that are contributed by females. So this then gets us to the whole idea of sexual reproduction. It just involves the combining of two haploid gametes and producing initially one diploid cell. And then that diploid cell will continue to divide via the mitosis process and grow and increase in size until you make a whole new individual. So I kind of worked up just a simple illustration of that life cycle. You can see it right here. So an adult individual is diploid, and so this is kind of the universal symbol representing diploid, 2n, right? Where n is a variable that represents what is the normal number of chromosomes in a single set, where you get one copy of every gene. All right. So the n number is just how many chromosomes in a single set, and then diploid, of course, would be twice that, 2n. So in order to make gametes, okay, there is a cell division process that is separate and unique from mitosis. We call this meiosis, and that'll be the subject for most of today's class, and we'll look at the mechanism for how that works. But by the end of meiosis, you end up with gamete cells that are N. N means haploid. And then you take one gamete from an individual, and take a separate gamete from a second individual, you fuse them together through the act of fertilization, and then we get our first cell is something called a zygote. And the zygote is a diploid. And then our diploid cell uses mitosis to grow and develop into a new adult individual, and the cycle can now repeat. All right, so that's kind of the context then to understand why we're studying this process of meiosis. Now, meiosis is all about second here. Okay, quick question. What chapter slides is this? This is chapter 13. All right. Okay, so let's move on, and we're going to now take a closer look at the process of meiosis itself. That is, how are gametes made? Right? And the interesting thing is that the evolution of the process of meiosis came out of the process of mitosis, and so there are a lot of similarities in these two processes. And so we are going to be using a lot of the same terminology. We'll be talking about monads and dyads. We'll be talking about spindle fibers. And even the basic stages or phases of this process are essentially the same. So meiosis has its own versions of prophase and metaphase and things like that. All right. So to give you kind of a quick overview of meiosis, again, this is the way those haploid gametes are made. And the only thing, though, is that because this evolved out of the process of mitosis, it does carry, unfortunately, a little bit of the baggage of that, particularly the baggage of doing DNA replication during an S portion of interphase. And so even before we begin doing cell divisions to make our egg or our sperm, each parent cell has doubled all of the chromosomes, and those will then condense up into monads, okay, and then, or really condense down into dyads, each consisting of two monads. And so there's going to need to kind of pull those monads apart from one another. Otherwise, if we just do a process of separating the homologous pairs of chromosomes from each other, then when the chromosomes decondense back into chromatin, we still have double the amount of chromosomes, which is something we're trying to avoid in making the cells in the first place. And so there need to be kind of two consecutive phases of cell division and literally separate forms of cytokinesis, so that the first division is the one where the cell manipulates the chromosomes in such a way that the homologous pairs can be split and separated from one another. And then once the homologous pairs are no longer in the same cell, then we can go through and we can separate monads from one another. And therefore, as kind of a basic level, one thing that makes meiosis a little bit different than mitosis is that in meiosis, we'll take one parent cell and we'll do 2 rounds of cell division, split the first cell into 2 cells, and then those 2 cells will be split into 2 more cells. So fundamentally, you can end up with 4 daughter cells at the end of meiosis, whereas back in mitosis, if you recall, we only had 2 cells. So specifically, again, we can kind of take a look at this illustration right here, and this kind of summarizes the big picture of meiosis. So here's what we're dealing with. We have a cell during interphase, and just to clarify, they oversimplified the illustration, and they're showing you cells in interphase where they look like individual monads that are coiled and condensed and even narrowed in at something representing a centromere. In real life, they don't look like this during interphase. Right? They only look like visible rod-like structures when you get into the first stages of mitosis itself. All right, but beyond that little quirk here, everything else kind of follows along. So here we are kind of during S1. You will have 2 individual chromosomes that are part of a homologous pair. Then you go through the S phase, and then by the end of S and into the G2 phase, now our homologous pairs each consist of sister chromatids that are actually loosely joined by a centromere. And then we proceed into our first phase of meiosis, what we simply call meiosis 1 with a Roman numeral. And through the subphases of meiosis 1, we figure out how to organize and sort and separate and then split the homologous pairs from one another. And they end up in 2 separate cells. And then meiosis 2 is, again, taking a look at, well, if every chromosome is made up of a dyad, we need to separate the monads from one another. And so we need a separate round of cell division to pull these apart from one another. And so here again, we see that fundamentally we can end up with 4 daughter cells by the end of meiosis. All right, so that's kind of the overview. So let's go through and let's kind of walk our way through the details here. Now, the mechanics of meiosis in terms of working with nuclear membranes and making spindle fibers and all of that other stuff, again, these are the same things that we talked about in mitosis in the last chapter. So essentially the process is going to work out the same. So I'm not going to really focus as much on the mechanics of how all of this works, and instead we're going to take a look at aspects that make meiosis a little bit unique and different from mitosis. All right, and then just try to combine the information from chapter 12 and apply some of that same knowledge here to chapter 13. All right, so here's our similar drawings like we had in chapter 12. So again, taking a look at the cell during interphase, you have an intact nuclear envelope or nuclear membrane. All of the chromosomes are in their spread out and loose state known as chromatin. And if this is an animal cell, like in preparation to make an egg cell or a sperm cell in a human, there would be pairs of centrioles that begin to organize the whole spindle apparatus. All right. And so those have been duplicated during our G2 phase of interphase. And if this is in the types of cells that are found in the reproductive organs of our organism, then they're going to get the signal to begin the process of meiosis. All right, so where do we go from here? All right, so meiosis 1, again, stage where homologous chromosomes are going to separate from one another. We have a prophase 1. And so just like prophase of mitosis, we're going to do a lot of the basic things here. We're going to begin to dissolve the nuclear membrane, the spindle fibers, the microtubules start to be assembled, and our centrosomes, with our centrioles in the center of them, begin to migrate towards the opposite sides of our cell. All right. So a lot of that's pretty much the same. And you also notice that your book here doesn't talk about an equivalent to prometaphase. And again, this is something that I mentioned in the last lecture. There's a little inconsistency even in your book on how to label the phases for cell division. And I found it odd that they use this new 5th phase, prometaphase, in mitosis, but then they don't talk about the equivalent of it in meiosis. Just shows to me a little bit how arbitrary the labeling kind of is. And so we'll consider all of the events of prophase and prometaphase of mitosis all happening here in prophase 1 of meiosis. All right. So that's all kind of the same. But then there are two very important differences that happen in prophase 1 of meiosis that we don't see happening in mitosis. Right. And that has to do with, first, if you'll notice that we have these blue and red dyads are all very, very close to one another and kind of aligned together. All right. First off, why the color scheme? All right. Well, the colors, again, represent chromosomes that were inherited from different parents. So we could say, for example, that the red chromosomes, the red dyads that you can see in the picture here, that those came maybe from the mother. And then the blue ones were inherited from the father, that kind of thing. It's a little arbitrary. You can decide if you want to attribute the colors the other way around, be my guest. It's, just saying that one color comes from one parent, the other color comes from the other parent. And these are what were inherited during the sexual production of this individual. Right. So why are they coming together? All right, well, actually, let's kind of close in. I've got a different graphic here. I'm going to switch to. Okay, this is an important aspect of meiosis, is this coming together of the homologous chromosomes that are a matched set. That is, again, they're the same size, have the same position for the centromere. And this process is referred to as synapsis. Right? And when this happens, we now form kind of a new sort of a unit that is going to get manipulated and moved around in the cell. So this unit is referred to as a tetrad. Right? It's a tetrad because now we have a total of 1, 2, 3, 4 monads that have all now grouped together. And so we call it a tetrad, basically, from the Latin prefix tetra, which literally means 4. I don't know how many of you are familiar with kind of the old school computer game Tetris. Or that's the game where you had all those different shapes that were made out of 4 different blocks that were arranged in different configurations. So why was it called Tetris? 4 blocks in every shape. Aha. There is methods to people's madness, isn't there? All right, so we have a tetrad. So this is sort of our grouped up unit that happens here. So that's one of the things that makes prophase 1 in meiosis different than prophase of mitosis, because we don't see the formation of tetrads in mitosis. Right. The other thing, and we'll come back and talk about the significance of this a little bit later today, is the fact that when you get synapsis and when you get monads from different chromosomes come close to one another, there is a little overlapping of those monads, creating a point where they cross over. This is referred to as a chiasma. All right, And the chiasma actually represents a place where there is going to be a physical break in the monads. And there is going to be a swapping of the pieces because each monad breaks in the same location, and then the fragments that were broken off are switched to the opposite monad. And so the result of this is that you now have some monads that now have a mixture of genetic information from both parents. So you can see down here where, I mean, yes, it says that we won't see this, won't be able to really appreciate this until you get to metaphase 2. And we'll come back to this in a little bit. But here you can see a monad that has red tips, but then the rest of it is blue. And then you see its counterpart in another cell, where this monad is mostly red, but with blue tips. Okay. So these types of monads that now have a new combination of genes. Originally, all of the genes that were on the blue chromosomes came from dad, and all of the genes that came from mom are on the red chromosomes. But now what you're doing is that you're doing a little mixing and matching of genetic information between the two parents, and now putting them on the same physical structure. And that's going to have some significance, so we're going to come back to that just a little bit later. Yes, it is very interesting. And the other thing I can point out too is that where those crossovers happen is different for every cell performing meiosis. Okay, so let's go back and take a look at the rest of meiosis 1. So we form tetrads, and some of the monads swap little fragments and pieces on them. And again, these can happen in different locations, and they can even happen in multiple locations along the monads that are in very close proximity to one another. The ones that are a little bit further apart probably don't engage in much crossing over as a result of it. All right. But once all of this is kind of being worked out, all the chromosomes continue to condense and tighten and become more compact and therefore easier to move. And then we're ready to move into metaphase 1. So in metaphase 1, we have connection between spindle fibers coming from opposite poles. And again, this is a little bit different because now that our chromosomes are arranged in a tetrad, all we need is a spindle fiber from one pole to extend just to one of the dyads. And then another spindle fiber from the opposite pole goes to the second dyad in the group. And so we're going to position, and then once those are connected, push and shove until all of the tetrads are aligned along the cell equator, and each dyad is kind of straddling the imaginary equatorial plane right here. And remember the metaphase plate from mitosis. So these are now kind of on opposite sides. Again, that's again a difference between that and the way it works in mitosis, because in mitosis, when you get to metaphase, all of the chromosomes are all aligned on the metaphase plate. But here now we have things split. All right. And then the real split is allowed to happen. Oh, so question. So how does this affect the identical twins? Are all of their chromosomes now the same? Well, Mara, if you're talking about after the crossing over, some of the monads are no longer twins. Okay, because now we've exchanged some of the genetic information. So let me go back to the previous slide here. All right. So again, just for simplicity's sake, all right, we can see that this monad, this half of the dyad that is a mix of colors, blue and red, this is no longer a twin to this all red one over here. Okay, and even I guess technically this one isn't even a twin to this one anymore, because now it's kind of a mix here. Okay, so now they're more like siblings. rather than identical twins. Okay. On the other hand, the monads that were not part of the crossover, they're still kind of the twins to one another. So again, they each contain all the genetic information from the respective parent that donated them. Okay. All right. So does that kind of clarify things? Oh wait a minute. I'm reinterpreting your question a little bit. So the idea of identical twins. Okay. If you're talking about are identical twins chromosomes all identically the same? Well, yes, they are, but that's only because identical twins form after the formation of a zygote and after fertilization of an egg. Okay. This is what we're talking about now in terms of meiosis. This is happening in the formation of egg and sperm cells before we get to fertilization. And so this part of meiosis doesn't have anything to do with the way identical twins have essentially identical chromosomes. Maybe that's a little more clear. All right. Well, throw me a line if you're still confused, and we'll try to come back to this when we get a chance. All right. So at metaphase, our tetrads are aligned up along the metaphase plate, and then we can move into anaphase. In anaphase, it gives the literal migration now, but instead of splitting, okay, good. We got everybody good. All right. I have random people joining our stream and cluttering the chat with things that don't belong there. All right. So where were we? Okay. So anaphase is the migration of dyads to opposite sides of the cell. So this is going to use, again, the same kind of mechanisms you talked about in mitosis. Either there's going to be a reeling into the microtubules from the centrosome, or we're going to walk the dyads along the microtubules using sort of the Pac-Man model. So, I mean, that mechanism is still the same here. It's just what are we moving? Well, here now we're moving dyads apart rather than moving monads apart. But this is the key event for meiosis 1, okay? Because this is what we want to do. We want to separate the homologous pairs from each other. And once we do that, now we can begin cytokinesis. We'll be able to split our cell in half. Now, one thing that if you look at this illustration here, there seems to be an implication that nuclear membranes are going to get reconstructed, and we're going to go through something akin to the normal telophase, like we saw in mitosis. Honestly, that usually doesn't happen because it doesn't make a lot of sense. Because we still have each dyad is made out of two separate monads that still need to be separated from one another. This again is kind of this is the leftover baggage from evolving this process out of the process of mitosis. We still have these duplicated chromosomes that need to be separated from one another. So it doesn't make a lot of sense to bundle them back up into a nuclear membrane because we're just going to have to tear it back down again in order to do the next phase here. Okay, so we have a question out there. It says, how do cells connect and conjoin? I'm going to have to get a little more clarification on exactly what you mean in terms of how do multicellular organisms, or how are they physically held together? Okay. Okay, I'm a little lost on that question right there. Again, that's really not a concern at this point in division, because if anything, we're trying to split cells apart. We're not trying to keep anything conjoined necessarily together. Okay. Okay. So anyway, so this kind of illustration of a telophase 1 really doesn't do a lot, and it's sort of a kind of just sort of a holding stage right there, because we still have more to do. So this will set the stage now for meiosis part 2. All right. So in meiosis part 2, now things get to be a lot more like our study of mitosis, because now the problem is the same in mitosis. How do we separate our duplicated copies of all of our chromosomes? We've separated the homologous pairs, but now we've got to get rid of the dyads and split them down into monads. So here's where things become a little more similar. You have a prophase 2, and during prophase 2, all we're going to do is make new sets of centrosomes, including centrioles if this is an animal cell, and start constructing new spindle fibers, making more microtubules. Again, they're showing fragments of a nuclear membrane that maybe had been constructed, probably wasn't, and so we don't really need that. Okay, so then we move, and the later parts of prophase, the spindle fibers now connect to our dyads and begin to position and maneuver them, so that by the time we hit metaphase 2, all of our dyads are now aligned along a metaphase plate, and each monad is connected to a separate spindle fiber going to opposite poles. All right. So we see that again in both of our daughter cells kind of happening at the same time. And then once those are all nice aligned and worked out, now we can have anaphase 2, where the monads are pulled to opposite poles, again using either one of the mechanisms, either reeling it in or walking the monad along the microtubule, getting them nice and separated. And then once those are separated, we can now begin our final cytokinesis, and we can begin kind of a true telophase and actually allow the chromosomes, our monads, to decondense and build new nuclear membranes around them, and we can break down any remaining spindle fibers and finish cytokinesis, and then that will complete our division. And so hopefully, in most cases, we end up with 4 daughter cells. All right. So that's kind of the mechanism. So again, let's just take a moment here. And again, let's just kind of compare mitosis with meiosis. So again, mitosis, again, we can divide up at very least into 4 distinct phases, where again, we start with a parent cell, we duplicate the chromosomes so that they exist as these sister chromatids, or two monads joined together forming a dyad. Right. And then get rid of a nuclear membrane, make spindle fibers, organize it all, and then get all of our dyads aligned up in the middle. Anaphase causes the monads to be pulled apart, and then repackage all of our genetic material into new nuclear membranes. And so the final result is that we have a daughter cell, 2 daughter cells. And notice along here is that we started with a parent cell that was diploid, because we had 2 of every chromosome. And we end up with 2 diploid cells as a result. So mitosis is all about maintaining genetic continuity, and particularly maintaining what we call the ploidy of the cell, starting with diploid, ending in diploid. Okay. All right, so question, are there any cases where telophase 2 does not split the two cells into four cells? Well, it's actually more like in cases where telophase 1 doesn't actually split the cell. Well, it can still split it into two cells, but then one of the cells that's produced is actually just a very small container of excess chromosomes that are not going to be needed. This is most commonly seen in the development of egg cells by mammals. And it turns out that when a human female, for example, is getting ready to release an egg cell during the ovulation phase of the menstrual cycle, the I lost my train of thought for a second. Where the egg is about to be released, it actually stops during prophase 1 and kind of holds itself into place. And it doesn't begin to finish the process of meiosis unless it makes contact with a sperm cell and it's going to get fertilized, which is really kind of strange. But then what it does is that it goes through the stages of meiosis 1 and it does a cytokinesis. But then what it does is that it takes one set of chromosomes, one of the homologous sets, and it just ejects it as a small cluster of just chromosomes called a polar body. Right. And then as fertilization sort of continues on and the sperm cell begins to penetrate the egg cell, it then goes through meiosis 2, and again it produces an excess set of chromosomes that have to be ejected. So the egg cells, duplicated monads or extra monads that we need to get rid of, those will be ejected as a second polar body. And so that egg cell kind of, as it went through all of its rounds of cell division, only produced technically sort of three products, but only one of them is legitimately a functional cell. The other two are just little membrane-bound sacs containing extra chromosomes, and they're not really functional cells. So I guess that would sort of answer your question is, do we not split two cells into four cells? That happens a lot in the production of mammalian eggs. All right. Okay, so back to our comparison here. Again, side by side. So looking at meiosis. So meiosis, again, we still start with a diploid cell. In this case, we had 4 chromosomes. Homologous pairs find each other during prophase 1. Crossing over also happens. Rather than aligning dyads in the middle of the cell at metaphase 1, we have our tetrads are all aligned. And then we have an anaphase 1, telophase 1, which splits our dyads apart from one another. And then we have meiosis 2, which again is almost a reworking of mitosis at this stage here. And then we end up with all of our various daughter cells. All right. So this is a really good graphic for you to spend time kind of studying. So again, you make sure you understand the similarities and the differences between these two types of cell division. All right. But let's get back to kind of our original problem. The original problem was trying to understand diversity and trying to understand what it is that's going on and why are offspring so different from one another. And particularly even when two offspring have the same parents, why don't they have a close enough resemblance to one another? All right. Well, this kind of variation is intimately related to the kinds of events that are happening in meiosis, particularly in meiosis 1. That's really what kind of sets the stage for making all of us just a little bit distinct and different from one another. And the main things that we're going to look at are functions like the independent assortment of homologous chromosomes. Come back and I'll explain exactly what that is. The random fertilization of gametes. All right, simply kind of the mixing, the fact that once egg and sperm are kind of out there in the environment, which sperm meets which egg is something of a random chance. And then we'll come back and again we'll take a look at this, what crossing over does for creating kind of new variations. All right, but let's start with this idea of independent assortment of homologous chromosomes. What does that mean? Well, simply put, it means that there is no defined system for organizing the alignment of the dyads across the metaphase plate during meiosis 1. Right? So what that means is that you can have a situation where theoretically all of dad's chromosomes align on one side of the plate and all of mom's chromosomes align on the other. And so then when we split this, open, we're going to end up with a set of daughter cells, a set of gametes that are going to have all of dad's chromosomes and all of mom's chromosomes going over here. So that, you know, you have gametes that have all maternal genes and all have moms. Okay. On the other hand, you've got another way of doing this, though. Okay. You can have it so that for one pair of chromosomes, dad's ends up on the left and mom's ends up on the right, but then you can have the reverse situation for another set of chromosomes. All right? And so what this does then is that this now separates, and now you've got chromosomes that have a mixture of dad's genes and mom's genes that end up in those gametes. And then you have the counterparts down here. So one of the things that this does is creates even new combinations of genetic information, whereas you can have some that are virtually the identical gametes to what came from one of the parents in terms of all the genetic information that came from this individual's father can now be passed on completely to the same individual's own offspring. But here we can now create a new combination of genetic material that is between the mother and the father at the same time. And as a result of it, okay, and we're going to simply have to see a mix. All right, so how many ways can you do this? Well, this is where it gets interesting. All right, all of the numbers here get really kind of fascinating, all right? Because what we've got is, well, let's put it to you this way. How many combinations of chromosomes can you put together? Well, it's really a function, it's an exponential function here. That is 2 to the power of n, whereas n is the total number of haploid chromosomes. All right. So let's just relate this to ourselves, okay? Humans, if you go 2 to the n, well, if our n number, our normal set of chromosomes in a single set is 23, that is half of 46, all right? Well, 2 to the 23rd power is approximately 8 million. All right? So in other words, in our own bodies, when we go to make either egg cells or sperm cells, we can have anywhere, any sperm cell represents a 1 in 8 million possible way of making it. All right? So there are that many ways of mixing all of the different chromosomes together. So that represents a lot of potential ways of making them. All right, just mixing mom's and dad's chromosomes, at least the ones from our own parents, and mixing those up. All right. Now let's kind of factor in this idea of random fertilization, the random meeting of gametes from one to another. Okay. this multiplies the possibilities, because if each egg represents a 1 in 8 million possible combination of chromosomes, and each sperm represents another 1 in 8 million possible combinations, then kind of the odds of putting those two together is 8 million times 8 million, or 64 trillion. And as a result of that, just dealing with. All right. All right. Sorry for the slight delay here. Okay, let's get back to the pointer. So the point here is we represent genetically a 1 in a 64 trillion possible ways of being made by our own parents. So this is why no two of us can be completely identical. All right? But it gets even better than that. Okay, so you're not just a 1 in 64 trillion possibility here. All right. Okay. This goes even more. Remember crossing over? Crossing over again was that during prophase, non-homologous or homologous chromosomes from each parent cross over pieces and mix with one another. And so the result here is combinations of genes that just don't exist in the parental chromosomes. All right. And as I mentioned before, those happen in different locations every time and at somewhat random locations every time. So as a consequence, again, you get this mixing in this production of what we call recombinant chromosomes. And every time cells engage in meiosis, they're going to make a different type of recombinant chromosome. And this is going to make them even more different than just the regular mixing of whole chromosomes, where you had that 1 in 8 million for humans times a 1 in 8 million to give you that 1 in 64 trillion. All right. And so the consequence here then, okay, is that you put all of these together, no two individuals are going to be exactly alike. It's just not going to happen. Okay, the only difference again is identical twins. And why do identical twins happen? Well, identical twins occur when you've already made a zygote. You've already begun to grow an embryo. And as a result of making that embryo, if that splits itself in half, and that can happen, is that you can take now clusters of this early person, and if it gets split into two separate masses, then both masses of cells can still continue to develop, but now they already have all the same genetic information. And so that's where we get what we call fraternal twins. These are the identical twins because the genetic information has already been figured out, because they started with the same original egg cell and the same original sperm cell, but now they begin to divide. The zygote has divided and grown into a new individual. All right. So that's the significance here. All right. And why does that matter? Ultimately, it matters because we have variation in organisms. This is the raw material for evolutionary change. All right. So this is where this was the one piece that Darwin never was able to figure out. Where does variation come from? Why are all members of a species slightly different from one another? And the main reason seems to be meiosis. Okay, and sexual reproduction is ultimately what causes individuals all to have those slight little differences. But then natural selection can work on populations and figure out which combination of traits are useful. Okay. Okay. So that's kind of where we're at. And I know it's a little bit early, but I'm going to go ahead and stop things now, and I'm going to get in here and I'm going to figure out a better way of blocking the spammers in our stream here and try to keep them from being such a disruptive influence. So I'm going to stop us a little bit early, so I do thank you for coming in. And then we will catch up with you in the next class. Thanks a lot, guys. I'll see you later.